Human beings have had cancer throughout recorded history. Some of the earliest evidence of cancer is found among fossilized bone tumors, human mummies in ancient Egypt, and ancient manuscripts. With the population of 1.22 billion, India becomes a major hotspot for cancer. According to the data estimated by National Cancer Foundation, India may face 1 million new cases of cancer every year with existing 2.4 million cases. Currently employed diagnostic techniques pose several limitations in terms of their limit of detection, high cost, lengthy turnaround times, intrusive sampling procedures, and requirement of a skilled operator. Among all these issues, cost comes out as the most crucial factor when we talk about the developing countries such as India, where affordability of the diagnostic test is a major concern.
In India, cancer adds to the already existing burden of non-communicable diseases. The need for early diagnosis of cancer is very important because it reduces the mortality, morbidity and the cost of treatment associated with cancer. Recent techniques which are available in India for diagnosis and treatment require a multidisciplinary approach, and it also requires a lot of specialized infrastructure. So in developing countries, that is sometimes out of reach of many for a large population.
In most of the cases, cancer is diagnosed at very advanced stages, stage 2 or stage 3, when it has been metastasized to the other organs. Cancer metastasis occurs when cells detach from the primary tumor, enter blood circulation, and begin to grow in distant locations in the body, a process which actually starts during the initial stages of cancer. Circulating tumor cells therefore can be a potential tool for early diagnosis and prognosis of cancer. Detection of CTC, especially in the early stages of cancer, is however a challenging task due to their significantly low concentration in peripheral blood and therefore requires technology with sufficiently high sensitivity, specificity and automation capability.
Detection of circulating tumor cells could be a promising strategy in cancer treatment because the detection and characterization of circulating tumor cells can help in proper diagnosis and treatment planning. These are the cells which are thought to be responsible for lethal metastasis and treatment failures in most of the cases. So if we can identify and characterize these circulating tumor cells, we can plan the therapy for cancer. Their early detection can help in early intervention and early identification of metastasis. This will finally add up to the improved treatment, increased survival rate and improved quality of life.
We propose a point-of-care device for simple, cost-effective and yet accurate detection of CTCs from peripheral blood. The device integrates bioconjugated magnetic nanoparticle technology for CTC enrichment in vivo with bioconjugated quantum dots technology for detection in vitro. Essentially, the first step is CTC enrichment in vivo by injecting a cocktail of magnetic nanoparticles with 5 different antibodies specific to 5 different protein biomarkers conjugated onto their surface. Permanent magnetic field is then applied externally near the blood vessels in order to concentrate CTC-bound magnetic nanoparticles in one region, thereby successfully enriching CTCs. Blood sample is withdrawn and isolated cells are subsequently permeabilized and stained for epithelial markers such as cytokeratin as well as for CD45 to exclude contaminating leukocytes. Subsequently, the sample is incubated with a cocktail of bioconjugated quantum dots with 5 different antibodies similar to ones conjugated to magnetic nanoparticles. This will result in CTC sandwiched between magnetic and quantum dots. The cell nanoparticle assembly is then subjected to magnetic precipitation, wherein it is kept in a magnetic field for 1 minute to precipitate out CTCs bound to quantum dots and magnetic nanoparticles. In order to prevent magnetic nanoparticle-induced quenching of quantum dot fluorescence, magnetic nanoparticles are freed using release buffer and free magnetic nanoparticles removed using magnetic precipitation. The suspension with CTC-bound quantum dots is then placed into a cartridge which goes into a laser-induced fluorescence detection system. The fluorescence of quantum dots is detected and correlated to the number or concentration of CTCs. The CTCs isolated by proposed technique can be further analyzed for the genomics and proteomics, and a more personalized therapy can be designed for each individual. The system can also be used as a point-of-care device to evaluate the success or failure of a therapy and the prognosis of the disease. The technology holds great promise in terms of market and commercialization potential due to its simple and cost-effective nature, and will benefit the large population of cancer patients in India.